The challenge of reliably expressing proteins has been around for decades. Because protein expression is a common activity in industry, there is a large driver for understanding the relationship between sequence and expression level. In recent years, the availability of GFP and the ability to synthesize gene-length DNAs at scale has led to a big data approach to the problem. I use this term big data loosely. As of yet, we aren't dealing with terabyte-scale data in synthetic biology, but we would if we could. The essential idea of these experiments is that a very large number of data points is collected, and then software is used to construct models that encapsulate the patterns present in this data. In this study, Plotkin and co-workers synthesize a large family of GFP genes and attempt to describe the observed variation in function in the sequence of each gene. Since the time these slides were made, a second and more detailed study has been performed by Church and co-workers. The conclusions are similar to the Plotkin study, but the refinement and techniques are more advanced. I recommend that you read both papers. In the Plotkin experiment, they synthesize 154 variations of GFP in which each variant uses different codon usage at each position. Thus, every one of these genes encodes the exact same protein sequence, but each one is encoded by a different DNA sequence. They place each synthetic GFP gene under the control of either a T7 promoter or a P-trick promoter. Even though all these genes have the same regulation and encode the same protein, there is a 250-fold range in fluorescence activity amongst the constructs. From a 50,000-foot point of view of this experiment, the most obvious take-home point is that the diversity of expression levels in prokaryotes is as sensitive to codon usage as it is to the upstream regulation sequence. Ribosome binding site libraries result in a very similar spectrum of activity as do codon variation libraries, and thus either route is a means of adjusting the gene expression levels in the cell. The Plotkin study goes much further in terms of trying to explain the pattern of expression levels as they relate to the underlying sequence of each construct. One very popular idea that is somewhat debunked by this study is that codon usage is a major determinant of expression level. When you examine genome sequences and tabulate out how many times a genome uses each of its 64 codons, you find that codon usage is far from equal, and it's different in different kingdoms. It is also well established that codon usage in an organism is correlated with tRNA abundance. For example, E. coli does not use the arginine codons AGA and AGG much, and this correlates with the fact that the tRNAs that decode these codons are poorly expressed in the cell. Anecdotally, if you overexpress these rare tRNAs in the cell, you often can improve the expression of genes containing rare codons. This suggests that the codon usage may be a major determinant of gene expression. The explanation for this would be that the rare codons slow the rate of elongation during translation, and thus the amount of product generated. The CAI is a value readily calculated for a particular codon sequence. It has the values between 0 and 1. It is defined as the geometric mean of the weight associated to each codon over the length of an open reading frame. Basically, it's a measure of the bias towards commonly used codons for any specific organism. It does not take into account the location of the rare codons in the sequence, but simply the count of each type of codon and the frequency of that codon in the host organism. Surprisingly, Plotkin shows that this CAI index does not correlate well with the observed expression level. They try to correlate the full-length CAI, the CAI for just the first 42 base pairs, or the number of really rare codons, and none of these statistically correlates with their data. This result is somewhat surprising, and superficially it seems to conflict with the frequently encountered observation that tRNA overexpression improves yield. One potential explanation is that slow codons are tolerated in some regions of the mRNA and not others, and we can only understand something like that by modeling the stochastic kinetics of the elongation process. Of the algorithms chosen, no one parameter predicts a majority of the observed variation. However, secondary structure in the 5' end of the mRNA turns out to be one major determinant. When they do secondary structure prediction to the entire mRNA molecule, that folding energy does not significantly correlate with fluorescence. If they limit the calculation to the first third of the mRNA, they see a strong correlation with fluorescence values. And this is most pronounced when the algorithm is applied to the region from -4 to +37 relative to the start codon. It could account for 44% of the variation based on T7 promoter sets. For the P-trix set, it explains 57% of the variation. So this region from -4 to +37 is the region that is encapsulated within the RBS calculator's delta G calculation, but it isn't altered by the design algorithm. They also examined predicted transcription termination, global GC content, length of the longest rare codon, and all explain some small component of the variability. Their study also looked at RNA sites. For example, the number of predicted ribonuclease RNase E cleavage sites predicts 4.7% of the variation. They also looked at the mRNA levels, and surprisingly, they have about 1/3 the variation of fluorescence levels. Considering that all these mRNAs are generated from the same promoter, this observation is somewhat surprising and suggests that translation and transcription may be more coupled than is commonly assumed. In general, the take-home point is that many different attributes of the mRNA structure end up influencing gene expression levels. Though the underlying mechanisms affecting gene expression all relate to biophysical parameters and conventional biochemical interactions which are well understood, it is the complexity itself that makes the problem challenging.